Okay, so like we were saying when I wasn't recording, but now we are, we might think that we were in some place like, I don't know, Ireland or Scotland. I'm trying to think of a place in the United States where this, but I really don't. I mean, the mountains kind of remind me of, the out west areas, but they aren't green like that at all. So maybe some areas in New England, maybe some areas I haven't been to, but regardless of where those areas are on Earth, they all sort of have a similar uh, climate, right? They're cool. We wouldn't think of this as being in the middle of the desert or being in a tropical rainforest. Tropical rainforest would have the rain. The desert would certainly have the sun, but the desert would have the heat, the dryness, and the tropical rainforest would probably have too much rain. And then there's the problem in the tropical rainforest is there are mosses there, don't get me wrong, but is the fact that there's the canopy, the big trees that grow, and they grow very thick. And I've never been. I would love to go there. I applied for a job. I applied for a job one time as an Amazonian river guide. So one of the requirements was I had to be fluent in Spanish. Well, the phone interview didn't go so well because I speak some Spanish, but I don't speak enough to lead a. I was like, oh man, would that be so great to go down the Amazon River and just, I would have to learn all this. I would just point to the things and call them their name in Spanish. Nouns are easy to learn. It was all the conjugation stuff. So I didn't get the job, unfortunately, but it's like one of those things out there. What you would get is just it gets very dark on the canopy floor. So the mosses that would grow there probably are just finding those little spots of light, right? So we would expect a place that would be maybe moderate sun, moist, like a lot of rain. Seattle, maybe, but Seattle's not that rocky. I lived in Seattle and it rains an awful lot. It's a beautiful city. They call it the Emerald City, emerald, like the green stone, because it rains so much that everything in Seattle is always green. All the plants are green, the moss grows, the algae grows. It's all very green, 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 green. But it is very pretty. Yeah, the only thing is the rain every day. It's like, oh my goodness, I know when I flew out there for a meeting many years ago, and as the pilot took off from the Seattle airport, he broke through the clouds. So we got up where it was sunny, and he made some comment about it, and something that people from Seattle rarely see. And when I lived up there for a year. Oh, and I can remember maybe a total of 2 weeks that I saw Yeah, it's beautiful, but man, I tell you, I am a sun person. I'm a sun worshiper. So yeah, no, yeah. Anyway, this is nowhere near where you guys are thinking. This isn't Ireland or Scotland. Well, no, it might have been Ireland, might have been Scotland. It could have been West Texas. It could have been anywhere. This is actually not Earth today. This is Earth 450 million years ago. All right, so this is Earth, what it looked like as we were discussing the other day, the evolution of the mosses, the non-vascular plants, the ones that have to grow very low to the ground. So I thought it'd be kind of neat, and that's what I've been doing this morning, is going finding pictures of what artists, oh my goodness, here somebody shot this on their camera. No, but it does look pretty good actually for an artist rendition. It's probably a computer, but whatever, I don't care. That's fine to me. I'm good with the illusion. But I thought it'd be kind of neat if we looked at some pictures and followed what was happening, since this is such a, I always say such an evolutionary driven course. And that'd be kind of neat to see what the environment was doing. So the first thing I would like to do, where's that one I want to see? What is this? Is this what I think it is? Yes, so here we are in the late Cambrian. This is where the continents were. The north is the north and the south is the south, but nothing else, looks familiar here. So this is what the Earth looked like continent-wise, during the Cambrian explosion. So that's when that rapid, evolution of life, and not only in the animals, but also in the plants. So picture, where we are geologically. So we got a lot of shoreline. I wish I could rotate this a little bit more because I really don't know what the underside, I'm sure the continents extended somewhat underside. We're probably only looking at the Western hemisphere, but nonetheless, there was a shoreline, there was areas near the shoreline, there was areas more further interior. Further interior areas would presumably typically be drier, right? Areas that, depending on how the sun, the latitude that the sun hits the Earth at the equator or higher up or lower down, that reflects how much energy, how much heat energy how much radiant energy is transferred to the Earth. If the sun hits it right as a round spot, then that's a concentration. But if the sun, is my pen working? Let's see. Okay, good. If the sun were to hit the Earth in one of the, now I know the Earth isn't tipped, so let's not throw that in there because then that becomes geometry and I get all screwed up on that. Other than getting thrown out several times from a geometry class that I didn't take to geometry very well. But anyway, so if the sun is hitting the Earth at this latitude, it makes this ellipse, right? That's not an ellipse, but you get it. And if it's hitting it at this latitude, it makes more of a circle. Well, an ellipse has a larger area, so same radiant energy, larger area, less energy per square centimeter, right? So less energy gets to the poles. That's why it tends to be cold and why the equator tends to be warm. Now, don't get confused if weren't the poles warm at one time because Antarctica was warm. Well, Antarctica was warm because it wasn't at the pole. Antarctica moved around with everybody else. Maybe, I don't know. Maybe this was Antarctica, right? Antarctica is a continent, but it was at the equator at the time, okay? That's why they find really cool stuff like tropical plants on Antarctica. You're like, whoa, that's so neat. All right, so there's the lake cam. What did it look like when we got a little bit closer? I found a neat picture of the Ordovician, and I need to make sure that I tell this to you in the right order. So give me 1 second to find something that cues me in the right. Yeah, all right. So here we go, Ordovician. So here's what it's like in the Ordovician, and what we see in the Ordovician is what I was talking about, that sort of increase in diversification of habitats. We got the really dry areas, but we got, these little plants down here. They're squeaking out a living. We don't have any trees. We don't have any bushes, but we got these little small things that could stay low to the ground. They were cool with diffusion and osmosis. They weren't bothered by the fact that they weren't too big, so they didn't need vascularization. It was kind of a rough place, so spores were probably the better way to go. Especially if you can make a covering that takes boiling acid to get off. You probably, and 250 degrees Celsius temperatures, things you're not normally going to find on Earth. I mean, there was a lot of volcanic activity still going on. Yeah, so given that was happening. So as the Ordovician went on, we have the age of the fishes. Oh, the Devonian. So we go, I'm sorry, no, let's not skip the Silurian. The Silurian, here's what's going on. Now, notice what's happening. Things seems to be cooling off. There seems to be more water vapor in the air. There's still some volcanic activity that might be warming the Earth. So look at the climate that we're getting here is tropical, right? Wouldn't you kind of guess a tropical climate? There are even some plants. The plants that you see in the foreground there, they're extinct. They're gone. That's called Cooksonia. I'll show you a picture of Cooksonia later on. Yeah, it looks like a Florida. They're taking the volcanoes away and taking the weird looking plants. Which, by the way, there are plants today that look like this. It's so cool. They're called fossil plants. Living fossils. I'm sorry, not of course fossil. Living fossils. A living fossil is a creature that's alive today that looks like something way back then that's gone extinct. It's like a convergent evolution thing, really, 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 really magnified. Okay. And I'll give you some examples of that as well. It also sometimes means something that we had gone extinct, but find out it didn't. Like that fish, the coelacanth, which, what an incredible find. Anyway, so here we are. And this is the Silurian. So this is about 450 million years ago. So this is the time when some of these plants that the mosses, they're still around. The mosses are doing their thing. But in the background, there are other plants, including these things that have gone extinct, that are starting to take advantage of a little better climate, right? Just a little better climate. Because before, where's my little clicker here? Hello, clicker. How come I can't go forward to, I'm on the wrong screen. Sorry, folks. Hold on 1 second. Why is... do it this way, then. It's not working. Oh, it's not letting me advance the screen. Let's start here. Yesterday, I couldn't do my afternoon class. It wouldn't let me load files or anything. It was horrible. Okay, so lost my me turn this off. Holy cow. Doggone it. Let me go back to that picture. Right? So we're cooling off. Earth is becoming more conducive for plant growth. Plant growth on land. Remember, plants were doing all right in the water. Even this time, animals were doing pretty good in the water. But I mean, come on now, the water is nice, and there are problems with the water. Small bodies of water, change very quickly. But if you're living out in a deeper part of the ocean, not too far out, but not too close in, you could probably find that Goldilocks place that's nice in the water to live. It doesn't change a lot. It's always close enough to have nutrients, that kind of stuff. But moving out on the land, that's a different kind of different circumstance. But it is becoming a little bit better. All right. So here we are in the Silurian, right? And then what I said. Now let's move on into the Devonian. Yeah, that's what I wanted to show you. Now in the Devonian, things become very nice. They become very lush, and things start to grow, and plants start to really take advantage of land of the different habitats. But there is a bit of an extinction that occurs in the Devonian. There's a drying out, or at least there are habitats that are starting to dry out. So what we're seeing here in the Devonian are plants that are starting to find different kinds of strategies to survive, which always is what happens when the habitat changes, right? So it's not that the entire Earth became this dry, sort of volcanically old, what it looks like, it looks like a desert. Those look like little tiny miniature cacti, don't they, growing there? Right, check out little centipede. I forgot, I didn't see him. Okay, but it's parts of the Earth are like that. So anytime there are different habitats, okay, you think about it, if a habitat changes, that's probably going to be more a gradualism type of evolution, right? I mean, relatively speaking, in the geological term, they might move in a million years. This mountain range was formed or something like that. It's probably longer than that. But when it comes right down to it, if the habitat is changing that slowly, organisms, it's probably going to be more like the gradualism model. But if it were to change pretty quickly, and that's a relative term, you're probably going to get the punctuated equilibrium. You might also get the punctuated equilibrium as the habitat changes, as it becomes various. Okay. You see what I mean? As the continent begins to form at the end of the Cambrian, what you really did at that time was you set up a candy store of habitats. You said, oh, you want to be an ocean dweller, deep ocean dweller, a shore ocean dweller. You want to be a crawling on the bank dweller. You want to come out. There were things that oh my goodness, the DNA was like, woo, check all this out. We can make all kinds of cool stuff with this. You see what I'm saying? And when DNA makes really cool stuff, what DNA makes, right, is organisms, right? Different types of organisms. And so that's why we get to just not to mention the billions and billions of species that have gone extinct, the ones that we have alive today, not to mention the species gone extinct. So again, let me return you back to this, because this is where we are when we were discussing the mosses. So we're at the time when the mosses have been around for a while, they're doing their thing, but other plants are investigating with other ways of life, and that's because the world is becoming various, right? Now, just as kind of a little bit of a hint, I found as I was searching for organisms of time periods, I found this one, which reminded me something I'm going to talk about when we get to the animals, but let me show you this critter. Okay, so imagine this habitat, right? So this is the plant landscape habitat during this time period. And at this time period, fish were doing pretty well. The Devonian is considered the age of the fishes. All right, so this is about that time, Silurian Devonian. And so the fish are doing pretty well, but there's that movement on the land. First with the plants, got to have plants first, but then some animals started to try. Look like this guy. Now, this thing is not alive today. The name Tiktaalik down below, or Tiktaalik. I'm not sure exactly how to pronounce it. I've heard it both ways, but I think Tiktaalik is one I've heard more often. But look at Tiktaalik, right? Tiktaalik is not alive today by any stretch. If you've ever seen something like this, please, because you can find every once in a while. We'll talk about the coelacanth more, like I said, but there's no Tiktaalik. So don't go out there be a Tiktaalik hunter. Don't be what they call a cryptozoologist. You know what a cryptozoologist is? Not real zoologist, by the way. What's a cryptozoologist? Anybody know? What's crypto? Not so much studying it. That would be more like a paleontologist might do that, or even just anybody in a particular, like an ichthyologist might study fish evolution. But the word crypto means code. Okay, a cryptozoologist is someone who spends their time chasing Bigfoot, Loch Ness, the Yeti, sightings of organisms like this one that have gone extinct, for hundreds of millions of years. Oh, somebody down in South Amazon found a Tiktaalik. And so, everybody rushes down there. Some cryptozoologist determines that this is Tiktaalik. My goodness, they didn't go extinct. And then all the zoologists look at it and they go, no, that's a blah, blah. That's a lungfish. That's an animal that exists today. You're just taking the picture and kind of distorting it, right? Crypt. Well, sure, because a crypt is secretive, right? Wouldn't a crypt be secretive? Even if you're buried in a crypt, it's kind of just you. Okay, but so can you imagine in that habitat I showed you with all the mosses that we were saying at the beginning, it was looking like Ireland. Can you imagine something like this having the opportunity, given the right genetic, having the opportunity to crawl, if you will, had to look at it, out of the water. But look what it's using to crawl with. Those are definitely not legs. Yeah, they're on leg. What are they? What would you say those are? How would you describe those? Kinda, yeah, that's how I would call it too. And check this out. It's even still got a tail with a fin. With a fin. Duck feet's another good one, yeah. Look at the head, though. Now look at the head. Where do you go there? Yep. And then look at the body. Check out those things right there, those little lines. You see those? What are those? Scales. So we got a reptile, flipper, I guess, holy cow, reptilian. No, amphibian, right? Amphibian. And then this could even be frog, a little bit like some frogs. Scales, which is definitely fish. Fin, which is fish, right? Look at, we have what we call those transitional fossil forms, don't we? We have evidence. Now, it didn't go a fish, Tiktaalik, and amphibian, okay? It went fish, less fish, more Tiktaalik, less fish, even more Tiktaalik, those ones that we sometimes, the puzzles that we're trying to put together. Okay, but you can certainly imagine in a habitat that we're talking about, an organism like this could have, I could imagine, if I walked down to that habitat or something similar to what we saw, at the beginning before, I would expect to find these crawly things, these things that could crawl out onto the mud and maybe get into the wet grass, salamanders, frogs, things that we find today, right? Wouldn't you all agree? Sure. All right, cool. Okay, so, there's kind of the framework as to where we are. Let me make sure I get back to my right slides now, because let's get into the lecture. So that's kind of what's going on Earth. Oh, I think I had one more. Oh, yeah, here we go. So we finished this all up, and this is where the Earth is at that time. Okay, so this is where the Earth is at the just about the end of this time that we're going to be talking about here. Okay, everything's coming together. Now it's not exactly in this spot. This is when Pangea forms, and my goodness, look at this. If you're talking about diversity, all right, if you're talking about diversity, look at all this inland diversity you have. Really far away from the water, not so far away from the water, kind of have a river going through you. Maybe you're out on an island. Now, one thing that does get screwed, by the way, is unfortunately the coastal area here, that decreases when the continent comes together. Look at it. If you got a bunch of little pieces, it's like that surface area to volume ratio thing. See that? You got a lot of surface area in a unit of volume if it's a little piece than versus if it's a big piece, don't you? You can do the math, but you remember that. So what happens is the organisms that live on the coast, and that's where most marine organisms live, there's not a whole lot going on out here, folks. I mean, there's phytoplankton and zooplankton, but, most of the productivity is going to be near shore where there's a lot more photosynthesis going on because things can grow, on the ground. Well, in the bottom, and because there's enough sunlight and there's a lot more nutrients. Things, unfortunately, sometimes bad things like fertilizers that run off the shore, right? So, but inland, holy cow, it's a party. Look at this, man. It's a party. You got dry stuff, rivers running through, et cetera, et cetera, et cetera, right? So that's the last one of that. Now let me show you your lecture slides. Sorry, like I said, I've been up this morning and I just started pulling up a few images just to kind of remind myself. And I'm like, oh, with the way people can do art now and computer graphics, and as much as we know now, it's just so cool to kind of do that. Wow, what did it look like back then? I'm always kind of doing. I've always been kind of doing that. I remember when I was a kid, and my dad, he was in the army. We're stationed in Greece, and so I went to the what's that big thing, the Acropolis. I remember just kind of standing there going, I wonder what it's like to some old Greek to be standing here looking up here at this thing, All right, so anyway, here we are at, 450 million years ago. So we've moved out of the mosses, the liverworts, the hornworts, those non-vascular, living low to the ground. Diffusion is good for me. Osmosis is good for me. I'm going to have a little weak, wrong word, a delicate gametophyte. I'm going to grow lots of now, we get sucked in this idea that you got to be a diploid. You don't have to be a diploid. There are critters out there that can spend part of their lives, or maybe all their lives, as haploid. You got a set of genetic information there. Diploid gives you a backup. Okay. All right. But the thing about having this haploid thing is it's a gametophyte, and you can make lots of gametes. So we're now going to be moving into the vascular plants, right? So here we go. There's the mosses that we talked about. Big thing with the mosses, again, is they don't have any vascular tissue. There's no seeds. There are sporophytes, which make spores, gametophytes, which make gametes. It's a numbers game over and over and over again. What you're doing is you're trying to make lots of gametes, lots of spores, because you know that most of them are going to, I was just trying to draw a skull and crossbones, but that's awful. Oh, my goodness. There you go. It looks like a moose with a skull and crossbones. Anyway, so you know that most of those gametes are not going to survive. Most of those spores aren't going to survive. All right. I'm trying to think where I stopped 2 slides ago. And you're right. I'm thinking, whoa, slow down. I'm sorry. Let's go back for a second. There we go. We finished up with the idea of a hornworts. I still needed to talk about the mosses. That's what it was. I'm sorry. So let's go to the mosses. So, you obviously see where we're going now, right? With another group called the mosses. Now, mosses, look at the gametophyte. Did I take this picture? No, I didn't. I wouldn't take a picture that good. But this is a sphagnum moss, the moss that grows here in Florida. Look at it. The green things are the gametophytes. I like the yellow better. The green things are the gametophytes. The little wispy brown things, if you blow them up, the picture down below, guess what those are? What are those called? Now, it's wrote on there. They make the spores, right? Look at those. Those are the sporophytes. So whenever you see a moss, I've been looking and I haven't seen any. I'm not real up on which mosses sporulate at which times of the year. But the next time you see a nice bed of moss, look at it a little bit closely. See if you see these little wispy brown things. And if I see some, I'll try to bring some in, or I'll take you guys out on the nature trail and show you. There's a big old bed of it that grows behind that sand live oak as soon as you walk out there, that sometimes has them. All right, but when you do lab, you're gonna break down a little more of the anatomy of one of these sporophytes. Like the stalk that holds it up is called a sporangiophore. A phore is anything that contains something. In this context, it's going to be the stick, the stem part that holds at the very top a sporangium. See, these are sporophytes with a sporangium at the top, okay? And that sporangium is going to make spores in a capsule, right? Now, these sporophytes, like I said, they're not photosynthetic. They're not brown. You're not photosynthetic. But notice this one over here. This one is green. It'll eventually become brown, but it does photosynthesize until it starts making spores. Why not? Why not, while you're growing, at least contribute to the food before you start making the reproductive part? It's efficient. It's awesome, right? That little tiny gametophyte there, here's like one of them down in the lower right. These things aren't even really called roots, folks. Remember, that's that technical thing. They're called rhizoids. Rhizoids are just anchoring. They don't do any absorbing. They can't absorb. They're not vascular. See, roots do two things, right? For a plant. Cooper's making his bed. If you hear him scratching in the background, I'm sorry. Coop. No. All right. Anyway, so rhizoids hold roots hold the plant to the ground and absorb water and nutrients. Rhizoids cannot do the second. They can only anchor. All right. So we're still why? Because we're still non-vascular. We don't have any tubes. All right. Anyway, so this is a single gametophyte right there. And at the very top of that, this whole thing is also sometimes called a gametophore. And you're like, well, it doesn't really have a stick. Well, remember I said that phore also means you contain that. So the gametophore, which is kind of the whole thing, contains at the tip the gametangia. Okay? So this is a gametophyte with the rhizoids and all that stuff, and then the gametophore would be that green part coming up. I'm sorry to throw these gameta at you. And the top is where the gametangia are formed. Make yourself some flashcards and write down these words. Write down the definitions. And then there are two types of gametangia, the female and the male, right? Make sure you get those terms right. And nice is they don't change much they don't change really at all through the rest of the plants. Archegonia is where the female a gamete is made, so it contains the female gametangia and the antheridia for the male, right? Figure out some way to remember. Anther, I always think antlers, male deer, I don't know. Think of some way to remember which one is which. Okay, now notice, and again, we talked about this the other day. Where do you find mosses growing? Well, you find them in moist, warm, the sun's not beating down on them. Well, one of the reasons is because their sperm are flagellated, right? They got to swim through something, and you can't swim through air. Now the way they do it is they swim through little droplets of water, which probably is very, very slow process. Phore, and put it an O, Ophore, gametophore. And the gametophore, what is that? It's the whole thing, really. Okay, so the gametophore is kind of the whole gametophyte gizmo thing, and then up here, this is going to be the gametangia, and then I'm sorry. Back up a minute. The whole thing is the gametophyte. Sorry. The whole thing is a gametophyte. The gametophore bears the gametangia. Just like the sporangiophore bore the sporangia, the gametophore bear the gametangia. You're going to be like, oh, enough with the gamma. Just write these terms down and use your flashcards, or use whatever method you use to make sure you make that that visual connection between them. Any questions on that, since I kind of confused you a little bit? The gametophyte is the rhizoid and everything. Then we break it down from there. Okay. Anybody got any problem knowing that these aren't mosses? I've only said it a few times, right? Neither one of this is a moss. Even if we call it a moss, it's not a moss, right? The upper left we call Spanish moss, not a moss. It's actually a flowering plant. The one below is not even a flower, not even a plant. It's a lichen. We call it what? Deer moss, reindeer moss, which I think it's funny in Florida, reindeer moss. Okay, any questions? That's just kind of a, what do you call it? A public service announcement, so you don't put those in your field book as that. All right, let's take a look, because this is going to help you. Use this slide when you're in lab on Tuesday, because remember, I'm not going to be there for you. So use the slide to help remember where you are. You're going to see slides under the microscope. They're going to look very much like I pulled these pictures up here. I pulled straight off of the web of what you're going to see when you look through the slide. Now, maybe they faded a little bit or whatever, but this is what they're going to look like. So let's go ahead and start with those. What am I looking at with the one I pointed the arrow to? Well, there I'm looking at the male part of the gametophyte, okay? I'm looking at the antheridia. See, this is the antheridia. That's the male part, anther, remember? And the dark purple in the middle there, those dark, oh, geez. It's hard to draw on a slanted surface. Anyway, that dark purple area up there, those are sperm, okay? So in the male part, in the antheridia, or in the male gametophytes, and we got to go back just a little bit. There we go. In the male gametophyte, what you have are antheridia. Now, there's also a female gametophyte, okay? And sometimes that depends on the spores. The spores, there might be, if you will, male spores and female spores that give rise to these, or sometimes, like we'll find in ferns, it's all different. I don't want to confuse you any more than that, but anyway. So we got a male gametophyte, female gametophyte. Okay, there's that gametophore again, right? Gametophore is the thing standing up. Gametophyte's the whole thing, even with the rhizoids. See? No tracheids. That's why they're not roots. In the female part of it, we have the archegonia. Now, under the slide, you're going to see something like this that you see right down there. I keep saying this without drawing. The archegonia, when you zoom in on it, looks like a vase to me. Okay, it's not really a good. It kind of looks like a vase. And if you look down the bottom, there's usually like a darker purple. That's the egg sitting at the bottom of the vase. Now, when they prepare the slide, sometimes the vase gets broken, you know, or something like that. And sometimes you look at them and there's nothing in the bottom. It might not have an egg. But look for something up in here that looks like a vase. It'll be obvious. The guy who's the lab tech, he knows quite a bit about kind of everything. So he might be able to help you with that. But if not, use the pictures there, or simply go up to the computer and type it in. Type in archegonium. Type in. You're going to be look. This is actually specifically going to be moss, or hey, write this down, by the way. You're going to be looking for moss, and moss is neem. I don't know if I told Scott, but when you're looking on the slide, it says one or the other. I can't remember, but they're the same. Okay, so the egg doesn't get dispersed. It stays in the archegonia. The sperm swims to little droplets of water, finds its way into the goes down that vase, finds the egg, and bop, there we go. We got Junior. Okay, and that happens with the archegonia. Now, what grows out of there is not going to be a separate individual, right? We're not doing it like animals do it. What grows out of there, there's a zygote, same as we got. Zygote is only when you put N and N together. That's it, right? And it grows as a young embryo, and so on, so on, and so forth, right? Now, you can imagine how delicate this whole process is. Now, notice that the young sporophyte, the brown thing, as it's coming out, the gametophyte is still haploid. The green thing is still haploid. As it's coming out, it's staying inside the sporophyte. Notice in the next picture, it never leaves. It grows out of the middle of that sporophyte. So it grows out of the middle of that vase, and it always remains nutritionally dependent upon the gametophyte. It never breaks free of the gametophyte. And it forms for one reason and one reason only, and that's to, bam, go make spores, right? Now the CD, here's another name for that little thing that holds up the stick there part is called the CD. Sorry. And you'll see this, why do biologists do this? You'll see that word CD or something that's pronounced exactly like it at least a few more times in animals. In general, it means things that stick out. Okay, so there you go. You can use that. At the top is the capsule. Look at this picture of the capsule I found. The peristome is the lid. The lid, right? As I said in the next one, when the peristome opens, all the spores come out. Look at all the little around this one, though, down here in the bottom. Look at all the little teeth. Why do you think the little teeth are there? I just noticed that this morning. I was like, oh, my goodness. What do you think the little teeth serve? What purpose? Now, visualize your garden hose and the different settings on it. That's what helped me. I was like, oh, I get it. Yeah, right? It sprays out. Like if your garden hose, you can adjust the thing, and if you have something that's try to be physics here, holy cow, trying to interfering with the water flow, it's going to break it up and make it very turbulent and make a spray, right? Now, if you don't have a spray, you have, clear or, whatever water. Yeah, so it's dispersal. And dispersal is important, isn't it? And we talked about that the other day. You're dispersing spores. Look how many. Yeah, I don't know if you can see the number. 50 million. Up to 50 million spores come out of one sporangium. And if you look among a bed of moss, there's going to be dozens, I don't know, maybe hundreds of sporangiums. So do the math. You're obviously trying to hedge your bets, aren't you? Now spores, the thing about spores, and the whole reason the sporangium forms is because the spores are created through meiosis. Bing, bing, you get your variability there. There's where your variability. Now you take your variability, and then mitosis can occur as well, because once you get a lot of variable spores, then you can really increase your chances by making lots of them. You don't have to necessarily make them variable. You might have, several thousand or million varieties out there, and then you just start cranking them out like on a copy machine. Some of them make them, most of them don't. If they make it, they become this little thing called a protonemata. Protonemata, very, very small. It eventually turns into a very scientific name called the bud. Actually, the bud is the apical meristem. Remember, the part that needs to know up from down. It's only one cell layer thick, but it already knows that's the way I need to grow. And that happens on the male and the female. And of course, we're back to the beginning of the cycle again. You guys have questions on that? I know I went through that kind of quick. There's some really cool videos. I think I put some on Blackboard. I don't remember. Check on Blackboard. If not, always go on YouTube and type in moss reproduction. There's really good, some's garbage, but there's really good animations and videos on there. You're not limited to those ones that I put on Blackboard either, right? It's not like you're like, oh, I better watch this. You might test me on it. I'm just kind of helping you visualize it. The big thing is getting all those terms down. Just remember, anything that has gameto is going to be something to do with the gametes. Yeah, I think they do, Thomas. I'm a very dynamic person. I need to see, and that's why I love talking about the evolution of life on Earth, because I like to see all the pieces, how they fit together. Unfortunately, you have to learn the pieces and before you can put them together, but I'm always impatient in that regard. All right, so anybody got any questions on that before I go on? Because I'm now done with the mosses. Okay, now I'm done with the non-vascular plants. We still got no seeds. We're still going to be dealing with spores. Seeds are very special, very special. We're not going to talk about those till the next chapter. So let's go on now. Let's go on now to these guys. Let's go on to the vascular plants. So the mosses, the non-vascular, the diffusion lovers, the osmosis dealer withers, all those, they dominate for a long time. So I don't want you to think that these things just, they never die off. They're still around today. But they're not flashes in the pan. Remember, life isn't a chain like Aristotle thought, or he called it a ladder. It's a bush. It's a bush, right? Everything's trying many different ways, and a lot of it's failing, and some of it's making its through. So the mosses had their day. It lasted 100 million years. But guess what was happening during that time again? It was getting drier, wasn't it? Or if it wasn't getting drier, there were areas that if you could learn how to deal with the dry climate, you could exploit things. If I could just move a little bit close over there where there's a little less water, there's a lot less, many of you, and then I ain't got to share sunlight or nutrients with or whatever it is that they're doing, okay? So as these dry climates became available, these vascular plants, and now vascularization is moving liquids around. So it makes perfect sense that at a time when dry climates became available or necessary, depending on how you live, where you lived, or how you lived, I should say, and then you had to develop some way to get that liquids out of the ground, right? You had to. And it's at this time that the sporophytes, they start to take over. And I don't mean that in a behavioral sense. The gametophytes are working just fine. And again, don't ever think that these things are failures, because if they were failures, even if the species isn't around anymore, the concept, the haploid, diploid generation dominance thing, it's still around. That's why mosses are still around. You walk around, you look at mosses, and they're still doing the gametophyte dominant thing, and it's working for them, but they're limited to habitats, of course. As the habitats change, other organisms have to find other ways, and being haploid is good, but if you have two sets of chromosome information, you got to back up. And if you're living in an environment that's a little bit more demanding, a drier environment, a harsher, whatever conditions you want to think about, if you're living in that kind of environment, it really helps to have two choices. Now, of course, when you're born, one of them's dominant, one of them's recessive, and there's nothing you can do about that. But if you happen to be born the other way, and it's an advantage, and I don't think I have to go on any further, right? Everybody knows where I'm leading to with that. That's what we've been talking about all semester long. So we have these plants around. This is Cooksonia. There's a fossil of Cooksonia. It's really kind of cool when plants fossilize because they don't do it very often. You know, remember I've talked about fossils, especially soft-bodied things. So that's what we think Cooksonia looked like. In the lower right is a picture I found online of someone who did a isn't that cool, computer graphic. And the things at the ends, well, those are the sporangiophores. Okay, so those are the sporangiophores. And when we mean that the sporophytes are becoming independent, what we mean is they no longer have to depend upon the gametophyte for life. Okay, they no longer are neutrally dependent upon her, or it, her, ooh, holy cow, on the gametophyte. So that means that those sporophytes now grow bigger and can grow stronger. And they're the things that are causing the variability. The gametophytes are just making the gametes. The gametes are very sensitive, so you're probably not gonna put them out in the first line of defense. You're gonna make these sporophytes. And maybe if it's a rough environment, you definitely want those sporophytes out front. But you're still kind of limited to not being very tall. You can't get very tall because you don't have any roots. You don't have any leaves. You got rhizoids. You can anchor, like this one in the picture. This one in the picture doesn't exist. It's extinct. Aglaophyton major. That's its stomata in the picture, by the way. Fossilized stomata. Isn't that cool? So that means we know that carbon dioxide levels on Earth, because when we start to see in the fossil record plants having more and more stomata, we know carbon dioxide levels are going up, because that's what they do with their stomata. Okay, it's that detective thing I've talked about. But you're still living pretty close to the ground. Okay. Everybody good with Cooksonia there? So there we are, at about the end of that period there where it's all lush and Ireland-like. We're now getting to the point where if it's not Ireland-like where you live, it's, it may be dry, it may be wet. If there's a million different, all the habitats we have on Earth today. Okay, so let's kind of almost seem like we jump ahead, but let's talk about the derived traits, the traits that came about, through the battle that ensued after all of this was going on, if you will, of plants that exist today. That's what we mean by extant, remember? Extant is the opposite of extinct. So when we look at plants today, based on the fact that they're still around today, and I mean, it's not, like I said, it's not failure of the overall scheme of life on Earth, but individual lineages fail, if you will, or individual lineages don't reproduce as fast, and of course evolutionary they die off. But if we look at the way plants evolved or the way they are today, we can get an idea of how they evolved. If we get an idea of how they evolved, we can often make very good assumptions about their environments, okay, and how that occurred. So let's do that. Let's start with one I just mentioned a second ago. Let's talk about this sporophyte thing. Right? Let's talk about this thing that is not haploid, which works, right? Check out bryophytes, the bryophytes, the mosses, right? Look at them; they're doing fine. They're still around today. But look at the gametophytes, little tiny wispy sporophytes. Have to depend upon the gametophytes. They're only around long enough to make other spores, and then they die after that. And then we jump over to these plants. We jump over to the tracheids. We jump over to the ones that can now absorb moisture from the ground and transport it throughout their bodies. Okay, so we have these organisms now that have the ability to grow larger, and when you can grow larger, you can start doing things like branching out those veins so you can make leaves. Now, if you're wondering what this thing is up here in the upper right, it's not a miniature anything. It's actually a fern. That's a fern down below. I'm sure you recognize that as a fern, but that's a fern gametophyte. That's how big it gets. And I don't have a scale for it, but they don't get very big. Centimeters big. I have ferns. Those are sword ferns, they look like. I've dug down below all the ferns I have growing in my garden out here, gently, like toothbrush and toothpicks, trying to pick around, see, and I've never been able to find one. So there must be some trick to it, I don't know. And if you're wondering what's growing out of the top here like that, well, that's going to become this part. And they're not called leaves, folks. And again, it's a technicality. Not leaves, but they're fronds. Okay, those are fern fronds. But if you hold up fern fronds, you can see that they're vascularized, right? They have these tubes running, so you can move stuff around. You can get bigger, and if you learn how to reinforce those tubes, you can get bigger. And if you can get bigger, you can overshadow these guys, right? It's all about competition. Holy cow, the sun ain't down, the sun is up. But of course, as I get bigger, there's the idea that I'm going to have to anchor myself to the ground. So see, it's all coming together here, right? All has to evolve together. So we get this idea of a sporophyte, the thing that can probably better deal with this much more demanding habitat, right? We use the sporophyte because the sporophyte has two sets of genetic information. Again, doesn't mean haploid doesn't work, but you're kind of limited. So the sporophyte's going to become dominant. Again, it's going to become, when you walk through the woods and you look at these tracheids, you look at these vascular plants, they have no seeds. Ferns don't have seeds. We're not there yet. Now, if you're thinking about seeded plants like flowers, they're tracheids too, but they just have those other things, which these guys don't have. There's no flowers on a fern, right? But when you walk through the woods, what you look at is the sporophyte. Whereas opposed when you walk through the woods with the bryophyte on the left, you recognize mostly the gametophyte. Sometimes, as I said, you don't even see the sporophytes. They're not there. Okay, let's talk about this tracheid thing. Let's talk about these tubes. Okay, they're called xylem and phloem. And here's how I remember from college. Xylem up, phloem down. That's how I remembered it. Xylem up, and I even had a visual because in xylem there's an X and a Y, and both of them together point up. Right, the xylem, they're tracheid cells. They're those veins that you see that are running through the plants. They're the ones you see in modern plants, the ones you see in ferns, the ones you do not see in mosses, though, because mosses do not have them. And as I said, the xylem brings up the water, brings up the materials from the ground. Okay, so now if you can add, tracheids in your rhizomes, in other words, if you can add roots, you can really have a head start on top of everybody else. Now, we're not quite there yet, but we will be there soon. In the lower right, there it is a picture, a cross-section to a tree showing you where the xylem is found. That's usually going to be the inner part. So we're looking cross-sectional, okay? So the water's coming, if you think about water coming up, it would be coming out of the page. And then the phloem's on the outside. And as I said, the water comes up. Oh, the xylem is also very lignified. So here's even a much larger picture. And again, you got xylem, different plant, phloem. The edges of those cells, those little tubes, the edges of the cells are lignified. And lignified is one of the most durable, organic substances on Earth. It's like rock. So it really lends a lot of strength to these large things that are growing. See, we're kind of getting large. That's what we're doing here. The phloem, like I said, that phloem down or phloem throughout, maybe. All the sugars, all the amino acids, the essential macromolecules, the essential organic molecules of life are transferred throughout the phloem. So you can see when they tap the trees up in Vermont to get the sap, see what they do is they cut into the phloem, don't they? They cut into the phloem because what the tree is doing in the fall is it's trying to take all these things that it's been making in the leaves, all these sugars and energy, sap is sugar, right? And it's trying to get ready to store it in the roots because it's getting ready for the leaves to fall off. And so it's moving, the sap is running down the phloem. So we cut into it and gather the sap, okay? All right, so the last part I've already talked about a couple times. So do you have any questions, right? It's all about out-competing each other, out-dispersing each other, because not only the taller you get, the more sunlight you get, but the taller you get, the further you can kick out them kids. Throughout, maybe. Let's think of throughout. Like when potatoes make potatoes, potato plants, the photosynthesis that occurs in the leaves, the starch, all that is stored in the potatoes, the tubers underground. So all that stuff that's made goes down the phloem and then creates those tubers. Now we can start talking about roots, okay? Now we can start talking about things that actually adds one more very important characteristic to plants, is that ability is to hold themselves to the ground, right? So let's take a look at this. As I said, roots by definition have those two things in red. They're anchors. So rhizoids are anchors or rhizomes. Okay, sometimes even today there are plants out there over on the right. There are plants out there that have rhizomes. But rhizomes are anchoring material. Okay, and you often see them confused back and forth. I know it's kind of like the same thing with the seaweeds, you know. They're called leaves, they're called roots when we know they're really holdfast. It's all a technicality issue. But to be considered a true root, you have to be able to do both of those things. And it makes sense because there were root-like structures, rhizoids, around before there was vascular organisms. Remember the bryophytes, the mosses have rhizoids, but a rhizoid can't conduct anything if it doesn't, you know, a solid piece of pipe can't conduct anything. It has to have a hollow tube in it. Make sense? What's interesting enough is we think, though, that the subterranean stems, the rhizomes, even ones you see today, I have a picture of one. I'll show you one. You'll say, oh, I've seen those. Here we go. This is St. Augustine. You recognize seeing that? You see that horizontal part, right? The long tube part that's got sort of a purplish color to it. That's a rhizome. Now, these organisms produce roots. It's St. Augustine grass. I'm not saying that, but what I'm trying to tell you is, let me get back to my slides again, that these rhizomes, yeah, everybody does, right? If you have St. Augustine, you do. You can't get rid of it. All right. Let's see, maybe go all the way back to that other slide. Let me do that. It's okay. Take a breather for a second. Vascular plants. There we go. True roots. There we go. All right, so those things exist today, but we're talking about the early organisms and even some organisms today again. The function of those little hair-like things, or those function of the, was just to hold on. And so we can see how they probably may have evolved from what we call this horizontal growth, these rhizomes. But one of the ways we can distinguish rhizomes and roots oftentimes is we say roots go vertical and rhizomes go horizontal. All right, so you see in the picture the rhizomes, and then down below it are the roots that are extending off the rhizomes. It could add anchoring to it. Of course, you can engage in nutrient absorption. But again, going back to the early plants, they only had rhizoids. They only had the ability to anchor themselves like you see in this fern. Now, interestingly enough, if we go back to the timeline, let's slide around. Let me jump ahead for a second. If we go back to the timeline, we think that roots are an example of convergent evolution because of both the lycophytes, which we're going to talk about, and the ferns have roots, and they evolved at different times. See, the lycophytes and the fern group had already split off. So that means it's convergent evolution, right? Because if that were the case, then we should see it in all of the ancestors of the split-offs, and it didn't. The gymnosperms and the endosperms have roots. All right. Where am I? Back here. Oh, yes, roots. Okay, so you understand what we got here. We got now roots. We have the ability to anchor and to absorb nutrients and absorb water from the environment. Okay, now the next thing that comes along are the leaves. And at this time, you can also imagine that on Earth, as volcanic activity is decreasing, it's probably a little clearer, right? Less stuff up in the atmosphere, less sunlight. If you can make advantage of less sunlight, if you got the genetics to do it, why not do it, right? So you can make these things called leaves. And if you're vascular, it's really not that hard to make a leaf. And there were two ways that it was done. Two ways that were done that today we can see in extant plants that are still successful. We can see among these organisms that we call the lycophytes that what they did is they did kind of a simple thing. Look over on the right. They made microphyls. A phyll is leaf. P-H-Y-L-L kind of refers to a leaf. And a microphyll is a small little leaf. If you look on that Crassus spike moss, those little tiny things sticking off are the leaves. And so what we have here is the vascular tissue, but not a whole lot of complication to it. See, it's one little vessel going up into the microphyll. So if you look down below to the origins, you can see how it come about. You had an outgrowth, and that outgrowth had vascular tissue. And then maybe what you did is you added some tissue to give it sort of a wing arrangement because you can capture more sunlight that way. None of that seems too unreal to expect to happen, and that has been successful for certain groups of plants. The Krause's spike moss today still exists, but think about this. Think about the change that could occur. Think about if you had these outgrowths. So go down below to the megaphylls, right? Big leaves. Think about if you had these outgrowths, these different branchings going out. And at one point, they were all, kind of like a bush. They're all going in different planes. But what would happen if somehow you had a mutation that occurred that all of those branches occurred in the same plane? So if I can hold my hand sideways, you can kind of see what I mean. If I have my hand in the same plane, I can then actually start growing tissue. I can start growing tissue. Ooh, I can do it with a green one. Cool. I can start growing tissue around there, can't I? I can start making a leaf. And the more tissue I can go around there, the bigger leaf I have. So like in the Tunbridge filmy fern, look, you can even see the vascularization there. Look at it, it's branched. See? And if you put a tissue, photosynthetic tissue between those branches, guess what you get? You get a big sun collector. And if you get a big sun collector, guess what you get to do more than anybody else? You get to reproduce more than anybody else. See? All back to the same thing we've talked about since the beginning of the semester. All these little innovations that add up. Things like the penguins walking 70 miles over the ice. Now, if we want to step back for a second and see where we are in the time period, we're at about 360 million years ago when the atmospheric carbon dioxide levels began to drop dramatically. Okay, now there are a few reasons why that occurred, but one of the reasons is the forest, the plant living things on land, they were becoming so doggone successful that they were actually having an impact on Earth at the amount of carbon dioxide. The water vapor, or I'm sorry, not the water vapor, but the volcanoes were depositing lots of carbon dioxide into the air. The respiring organisms in the sea, a lot of that carbon dioxide, not all of it, but some of it was going up into the air. The respiring organisms on land, and now you got these things crawling out on the land. And they're all respiring, giving off carbon dioxide, but you got a boatload of plants. The era that we're in, the name for the era that we're in is called the Carboniferous. About 350, 360 years ago, million years ago, yeah, 360 years ago. It's amazing to think of the time scale. We call it the Carboniferous. And the reason why we call it the Carboniferous is when you dig down through the rock layer and you get to the rock layer that's that old, guess what you find? Please don't say live plants. What do you find, you think, when you dig down to the Carboniferous 360 million years ago? When I was a kid, I was always going to threatened by my parents that I was going to get it in my stocking for Christmas. Now you know. Yep. Coal, right? Okay, so these atmospheric carbon dioxide levels began to drop 360 million years ago because of all these plants. So think of how much coal there is. They estimate another 400 years of coal on Earth. There's that much coal. There was a boatload of plants. That's why it's called the Carboniferous, because coal is black and carbon is black, right? So during this time when the atmospheric carbon dioxide levels were dropping, it would have been advantageous to make larger leaves. So we can imagine a situation where the megaphyll plants would have then begin to rise in terms of, say, species diversity or species number because of the fact that they could take advantage. It doesn't mean that the microphyll plants died. They didn't die. They're still around. A lot of them probably did, but some of them were able to survive. Another advantage of having larger leaves, remember I told you earlier about the Cooksonia with the fossilized stomata? Larger leaves, more stomata. It's kind of an Italian thing. Larger leaves, more stomata. More stomata. More stomata, more carbon dioxide you can suck in. There ain't a lot of it out there, so the more I got, the more I can suck in. But with everything there comes a price, doesn't there? For every innovation that organisms come up with, there's a cost to be paid. So if you're getting larger because you want to take advantage of more sunlight, sunlight is not just light, it's also heat. And you begin to overheat. And we know from Bio 1 that that's not good. And we know from Bio 1 that there are, oh gosh, numerous enzymes that are involved in photosynthesis and respiration. We certainly don't want to denature any of those. So what are we going to do about this overheating process? Well, these stomata that are growing on these larger leaves, they have the answer. Kind of, they have the answer. What they do is the stomata can open up and not only exchange carbon dioxide and oxygen, which is one of their purposes, but they can also lose water. And when water is lost, it's called evapotranspiration in a plant. It's sweating, folks. It's sweating. It's water evaporating off of a surface, and the physical process that says that removes heat from that surface. So evapotranspiration in plants is evapotranspiration, water evaporating off the surface of the leaves that cools them off. Now, as I said, that's a double-edged sword, right? Why is that a double-edged sword? If you remember from your Bio 1, you'll understand that's why there are C4 in CAM plants. Why is evapotranspiration bad as well as good? Of course, that's never good, right? Never good to lose too much water. Dehydration is good for nobody. So yes, you sweating is good for you to cool off in the summertime, but you can also dehydrate, can't you? Mother Nature does not leave any easy solutions. There's always a price to be paid. Okay, another thing that land plants did, extent land plants that we see that was obviously very successful, because we see it around today, is monkey around a little bit more with the sporophylls. For one thing, they started producing different types of spores. And they started modifying these sporophylls, these little brown wispy things, into spore leaves, okay? A flower, a pine cone, those things are really nothing more on the plants that we haven't talked about yet, nothing more than modified leaves. Everything on a plant is a modified leaf. As a matter of fact, a cactus, the spines on a cactus, those are the leaves. The big green thing, that's the stem, if you will. Cacti have evolved so that their stem is the photosynthetic part, and the leaves get sharpened into these nasty little things that I have great fond, well, not so fond memories of growing up in West Texas, of picking out of my skin. That's the leaves. So one of the ways you can modify your leaves too is you can make pine cones, you can make flowers, you can make fruits, and we'll talk more about that later on. But it's all about the sporophylls now. Those diploid, I can deal with a little more harsh than haploid things that were becoming more and more prominent. Okay, on these sporophylls, these specialized leaves, there are sporangia. Okay, look at the picture of the fern in the middle one. We showed you that the other day. The fern frond. The frond, not the leaf, the frond is the sporophyte. Okay, on the sporophyte there are sporangia. In a fern, they're called sorus or sori, plural. Look at all the sori on the back of that fern plant. Have you guys checked out your fern plants to see if you have any? I haven't checked out mine. I should go out today and take a look. Like I said, I'm not exactly up on when ferns sporulate, or mosses. Guess I should be. Check though. All right, go out there or any fern that you see, just flip it over. Now, not all ferns do this, but many of them do. So, okay. Now, on the other hand, the lycophytes and things we call gymnosperms. Okay, what they do is they produce not a sorus, but there's something called a strobilus or a strobili. It's plural. It's kind of maybe a difficult word to remember unless you remember that the Latin translation. So that's what I put that there. Latin, the strobilus was a cone. What we're talking about here are plants that produce cones. So immediately the top picture comes to mind, right? Cones. Those are pine cones. Pine cones on a pine. I took that picture, pine cones. Now you notice there are two types of cones, aren't there? What are the cones down below, or what's the difference between the two cones? Or the two types of cones? Nicole, Sydney, Marcelo. Jump in there, guys. What are those other cones? Why are there two types? That's their male and female. Yep, they definitely are a different shape. Absolutely, right? And that reflects what they are. That's the one down below is the male cone, and the one up above is the female cone. And there's a very good reason why there's a male and a female cone, just why there's a very good reason why there are male and female animals. It's all about diversity, okay? It's no different than the fern making a whole boatload of spores. All right, we'll talk more about cone plants later on. Let me talk about the other thing that happened with spores is the fact that they became variable. Okay, now among most seedless plants, which is where we still are, it's only the vascularization that's changed. We're doing with ferns, we're doing with mosses, we're still talking about seedless plants. Okay, among them, and I try to do my best to help you with terminology, so that's why I drew up this little chart here. Sporangium, spore-producing structures, are found on sporophylls. Those are leaves. Specialized. A pine cone, male or female, is a specialized leaf. Okay? May not look like a needle is a leaf. Okay? All right? So in most seedless plants, the sporangium on the sporophylls only make one type of spore. Typically, it's a bisexual, or it's a spore that forms a bisexual gametophyte. And what I mean by a bisexual gametophyte is on the same gametophyte. There's the gametophyte, right? There are the male and the female. There's the archegonia and the antheridium on the same gametophyte, on the same structure. Now, does that work? Obviously. Mosses do that, okay? Ferns do that. When you look in the microscope, but we'll get to the ferns today for reproduction, but when you look in the microscope next week, you will see on a fern, a gametophyte, the male and the female parts on the same structure. Now, does that cause inbreeding? Yes. Yes, it does. You can't prevent it. You can't prevent it. One of the things they try to do is one of them will produce a sperm or egg a little bit at a different time. I think it's the sperm, if I remember right, what I wrote down, the sperm are produced first. So they have a better chance of dispersing away from their own gametophytes before the eggs mature. Yeah, I think that's the way it is. The sperm produced first, they're liberated first. They've already spread out, hopefully to other moss individuals before the eggs mature. It works, okay? Oh, but the other hand, with seed plants, and we haven't got to those yet. We're still in that in-between vascularization, but still no seeds, okay? That's why I said a fern has vascularization, okay? Does not make a seed. Still a non-seedless. But on seed plants, we get what's called heterospores. And hetero means what it means in anything else. It means other, okay? So again, I tried to help with the terminology here by reminding you, or not reminding, but trying to make a visual connection in the red. The female gamete, even if we're talking about the female egg, is much, comparison-wise, much larger than the male gamete. A female's egg is, I don't know what it is in terms of proportion, but you've probably seen pictures of an egg with a whole bunch of sperm swimming around it, right, or stuck to it. You know that the egg is massive compared to sperm. So, excuse me. That's why I highlighted the word red, word in red, mega, okay? So here's the terminology in those heterospores plants. Megasporangium, things that make spores, are megasporophylls, a female pine cone, for example, okay? Produce megaspores, which produce a female gametophyte, the female pine cone, which makes an egg. Microsporangium on microspores or on microsporophylls make microspores, make male gametes, which is what's produced in the pollen cones. That's what pollen is, folks. Pollen is sperm. Pollen is sperm. Pollen is sperm. What it is. All right, and down below, before we leave, because I'm not sure I remember to point it out, there are the cones of a lycophyte. There are the strobili of a lycophyte. I can't tell you that I've ever seen a lycophyte in person, so I'm not sure how well I could describe it, but you could easily look it up online and find out what it looks like, okay? So you ready to start jumping in and looking at the different types of plants? This is exciting stuff to talk about because to me, it's just like I said, step by step, little tiny things. And I think about all of the different branches that were tried, even all the ones we don't know about yet, right? All of the different evolutionary lineages that went extinct. So let's jump right into the lycophytes, things called the club mosses. There they are, club mosses for reasons pretty obvious. They have clubs. The quillworts, which I think are cool. I'd like to see one of those. And spike mosses. Now, I'm not going to bet my paycheck on it, but I don't believe we have any of these in Florida. I'm not really sure. I put mosses all in quotes because I want you to keep in mind that, remember, these aren't really mosses. Mosses are bryophytes. Mosses are non-vascular. Some of these organisms are epiphytes, epiphytic. That means they grow on top of other organisms, like an orchid does. So yeah, moss-like, but not mosses. This is what the Carboniferous looked like. That was the other picture that I had. This is the period right after what I was showing you with the Ireland kind of picture. So the trees are getting big. The horsetails. Oh, we still have horsetails around today, but they're not that big. I'd love to see one that big. But horsetails, ferns, the lycophytes, these small little plants that are around today. Lycophytes, I don't know off the top of my head how big they get, but they're ground-dwelling plants for the most part. Back then, they were trees. Those trees in the backgrounds were lycophytes. Now, the homosporous and heterosporous stuff is starting to evolve about this time. So that's why in this same group, you get the club mosses, which are homosporous, and yet the other two major parts of the group are heterosporous. Right? Everybody's trying out something different. And again, if something did not work, it would not be here. So don't look at it as being heterosporous is better than homosporous. It just depends. And just when it depends in one way, it depends in the other way, because Mother Nature likes to do that. Let's move on now and talk about the monilophytes. These are the ferns, the horsetails. My favorite plant. No, ginkgo. Oh, well, anyway, I've worn one. And something called the whisk ferns. I've only seen a whisk fern once. Everybody's familiar with the ferns. The ferns are actually the closest group to the seed plants. So what I mean by that is right here. Ferns and seed plants here share a more common ancestor than ferns share with lycophytes. Right? Lycophytes and ferns had a common ancestor 425 million years ago. Ferns and seed plants 380 million years ago. So ferns are actually closer to seed plants, the subject of our next chapter. So it's called a sister group. So we have the typical fern. We have the horsetails. Oh, let me show you the horsetails that I found. Oh, we're good. Let me see here. We might run a little bit, but we'll figure it out. All right. Here's my horsetails. I'll have to show them to you next time. I guess I forgot to upload them. Anyway, I saw a bunch of horsetails when I was going down a trail the other day. I thought it was kind of cool. So they grow in ditch side areas here in Florida. I found them near an old, or not an old, but a place that grows palm trees for landscapers and that kind of stuff. The little tiny green things that are pointing up, those are the leaves. All right, very, very simple type of leaves here. Okay. And there's two types of stems too. There's the vegetative stem, which is all involved in photosynthesis, and then the reproductive stem, the strobolus. The strobolus that you see there, that brown thing, and I have some picture of those. I have to show you those in class. Those are the reproductive parts. I saw a whisk fern once down in Kissimmee. Oh, sounds like a title for a song. Growing over in kind of a ditch. I unfortunately didn't have my camera at the time, but you can see again it's a strobolus. The whisk fern is that living fossil. I'm glad I remembered that. That's the Cooksonia plant that I showed you very early on. The one that I said had gone extinct, but you're going to see something later on that looks like this. This is called a living fossil, not because it's not related at all to Cooksonia, relatively related, but it looks a lot like Cooksonia. Okay. And then the ferns. Yeah, that's okay. We'll go a little bit longer, and then we'll stop when it's time. We can pick up the rest later on. The fern's 12,000 species. Everybody knows a fern. There's big ferns, little ferns. That fern in the upper right, I wish I had extra semester to go through all this stuff if I really could. The fern in the upper right, I wish they hadn't switched combined bond in. So that's called the cinnamon fern. That's the reproductive part. They don't make sori on the back. If you flip over the frond of a cinnamon fern, you won't find those little dots. They make it doesn't always have to be the same way. They make another thing, another frond, if you will. It's called a reproductive shoot. Those look like sticks of cinnamon. I took this picture of a bunch of across the street. They're not always, they don't always look like this. But if you hold them under a microscope, you can see the little sori. They're all rolled up and everything. It's really, really cool. Most ferns have a bisexual gamete, right? Which means that their spores are homospores, right? That's what it means to have a bisexual gamete. You have one type of spore that gives rise to a gametophyte, which has both Antheridia and Archegonia. money, yeah, right. Numbers, right? Trillion spores in a lifetime. Very tall tree, the tall green thing, that's the very prominent part. That's the sporophyte. Very small. I showed you that little tiny weird looking thing. That's the gametophyte. The advantage is that they both can photosynthesize. So unlike we had mosses where the spore, these little tiny gametophytes in the lower right, they will photosynthesize, so they can help with that. Growing out of that thing again, growing out of the bee, if you will, when you're looking at this thing through the microscope, it'll be growing at you. That's going to be the frond. That's going to be the sporophyte. Okay, so stick with me. If we got about a minute, stick with me. Let me go through reproduction of the ferns. It'll only take me just another couple minutes if you guys don't mind. So let's go with the gametophyte. We're here. These are not roots. Okay, these are not. Or wait a minute. These are roots. The rhizoids on the gametophyte are not roots. Sorry, I got ahead of myself. I was looking at one thing and saying another. So the ferns are starting to produce roots, start producing vascularized anchoring points, but the gametophyte is still only has the anchoring points. Okay, so, and also horizontal roots. Rhizomes are becoming very popular, okay? So that's very good for vegetative reproduction. Grow along this way, and then you pop yourself up. These fronds are the sporophytes. They contain sorus or sori, many of them, which contain a sporangium. Now, sporangium makes spores, and spores are created by meiosis. So we're making variability, right? So there's the variability part. Whenever you hear meiosis, you think variability, and then we might mitosis and make lots of those spores, and then we might germinate them, like spray them out like the ends of a fire hose, put a spring on them, whatever, get them out there, okay? If those spores, it's only one type, it's homosporous. Those spores, one of those makes it, survives, it's going to form that little tiny young gametophyte. Eventually, that gametophyte is going to grow into this heart-shaped thing. And since it's bisexual, since that's a homosporous plant on the male and the female, and think about this when you're in lab, folks, the Archegonia is going to be up near the V part, and the Antheridia is going to be back towards the other extreme, back towards the other side of that. So Antheridia and Archegonia. And then, of course, the sperm are going to flagellate it. They swim through water, and they find an Archegonia, find an egg, and everything is hunky-dory. You got a junior. All right, last slide, then I'll stop. This is nothing more than pictures. This is another good slide that you might want to make sure that you have and can bring to school when you're looking at under the microscope, because it's the same thing. There's the Antheridia, there's the sperm, there's the Archegonia. Antheridia look like a soccer ball. Archegonia are looking down the vase. There's the egg at the bottom. Okay, now this is what it's going to look like on the fern, not on the mosses. All right, so here comes the developing fiddlehead. The fiddlehead, it looks like a fiddle, looks like a violin. It unrolls. The sides unroll, they produce sori, sporangia with spores. If they germinate, they make gametophytes that are bisexual. All right, so I try to get you lots of pictures to help. Remember, you can always, if you bring your laptop, by all means, search on there, search on the laptop at the front of the class, whatever you need to do to help you identify these things under the scope, okay? We have a couple more things to talk about, but I won't keep you any longer, so I'm going to go ahead and stop the recording here. Wait a minute, hold on a second. Do remember that you do have your exam. Somebody just sent me an email and I lost track. You do have an exam on Tuesday, right? Don't you? Yes, and you are doing lab 5. So we're only doing part of lab 5, not the whole lab. We're only do up through and including the whisk ferns. So we won't do the gymnosperms and the angiosperms, which is the last part. Okay? I will try to be on the computer as much as I can when I'm in this conference. We'll be able to occasionally check my email. If you have any questions for me or anything like that, by all means, give it a shot. I will do my best. I will do my best to return your email. Okay? Other than that, I'm going to stop the recording.